Okay, now that you have fully understand glycolysis, which is the initial metabolic pathway, let us now discuss Krebs cycle. So Krebs cycle is also known as the Hans-Krebs cycle, which was discovered by Hans Krebs, who has received a Nobel Prize in 1953 in physiology. It is also known as the citric acid cycle, since citric acid or citrate plays an important role in this cycle. We are going to discover why. So the Krebs cycle is the key metabolic pathway that connects carbohydrates, fats, and protein metabolism. This cycle is the central pathway in which many metabolizes. It consists of series of chemical reactions which generate NADH and FADH2, which in turn generates ATPs. So the Krebs cycle consists of series of chemical reactions that release stored energy or fats, carbohydrates, and proteins through the chemical process known as the oxidation. So aside from producing ATPs, it is also important since it provides many raw materials like amino acid and NADH. An interesting fact about Krebs cycle is that it is one of the earliest established components of cellular metabolism and may have originated abiogenetically, meaning it originated even before life existed. So the main concept of Krebs cycle, we need to discuss the main concept before indulging into the steps. So as I have told you, Krebs cycle is the central metabolic pathway. Central metabolic pathway. What do you mean by that? Okay, imagine when we eat food. It is digested, absorbed, and processed by initial metabolic pathway. And this initial metabolic pathway produces some common products that are used by the central pathway, which is the Krebs cycle, to produce energy. Example, the bread, it is a carbohydrate. It is digested into simple sugars like glucose. And the glucose is processed by initial pathway like glycolysis. So this glycolysis produces the pyruvate. And similar products are also produced by initial metabolisms of fats and proteins. So the pyruvate, the end product of the glycolysis, is then fed into the central metabolic pathway, which is the Krebs cycle. So this Krebs cycle provides a central pathway that connects carbohydrates, fats, and protein metabolisms. So that is basically the concept of Krebs cycle. So this is the process of the Krebs cycle. The Krebs cycle begins with a molecule known as the acetyl-CoA. But the question is, where does this acetyl-CoA came from? And what is the role of this in the Krebs cycle? So let us go back to the pathway that we have discussed earlier, which is the glycolysis. So in this pathway, we were able to break down the glucose, which is a 6-carbon compound, into 2 molecules of pyruvate, which is a 3-carbon compound. So we have the end product of glycolysis, which is the 3-carbon compound pyruvate. What happened next is that this pyruvate is acted upon by a set of enzymes collectively known as the pyruvate dehydrogenase. Very good. Dehydrogenase complex. So in this reaction, coenzyme A, which is a vitamin B5, is also involved. So what happened in this reaction is that the 2 carbon are transferred from the pyruvate to the acetyl-CoA. Transfer to the coenzyme A from acetyl-CoA. One additional carbon present in the pyruvate is lost in the form of carbon dioxide. So in this reaction, one molecule of NADH is also produced. So this is how the acetyl-CoA is produced from the pyruvate. So this acetyl-CoA is a 2-carbon compound and is the starting compound in the Krebs cycle. So this acetyl, or in this molecule, the CoA, the part which is derived from the vitamin B5, acts just like a carrier, which responsibility or duty is to transfer a 2-carbon compound attached to it to the Krebs cycle. So that is the job of this coenzyme A, which is attached to the acetyl. So let's move on to the first step of the Krebs cycle. So in this first step, what happened is the acetyl-CoA is combined with the oxaloacetate. So like I said earlier, acetyl-CoA is a 2-carbon compound. And this 2-carbon compound combined with the oxaloacetate, which has 4 carbon. So this combination leads to the formation of, anyone? Yes, the citrate, which is a, how many carbon does the citrate have? A 6-carbon compound. And this reaction is made possible by the enzyme known as the, yes, the citrate synthase. So in this reaction, we have here, let's show the reaction that happens in this. So in this reaction, with the help of the citrate synthase, okay, so there are molecules of carbon dioxide that is lost as a waste product of this reaction in the first step of the Krebs cycle. So there is a 10 reaction process which lost carbon dioxide, 2 molecules of carbon dioxide. So enzymes generate energies through oxidation. So the very process that we are going to discuss is the oxidation reduction reaction. So that is only the first step. Now, let's move on to the next reaction, the second step of the Krebs cycle. So in this next reaction, the citrate is converted in its isomer, which is known as the isocitrate. So this reaction is catalyzed by the enzyme called the, anyone? Yes, the aconitase is the enzyme responsible to this reaction. So this is an ion-containing enzyme which duty or which responsibility or function is to reorganize the atoms of citrate to form its isomer. But why does the citrate needs to be reorganized? Why can't the citrate move forward to the next step of the pathway? Well, the answer to that is because the arrangement of atoms in the isocitrate is better suited to the next oxidation reduction reactions that are about to come in the cycle. So that is the explanation why the citrate needs to be rearranged in the next step of the cycle. So, if dili siya i-rearrange, that means na ay problem na mahitabo sa next step. It's because they need to be corrected in the first step pa lang. Okay. So moving on to the next step, the isocitrate is converted again into alpha ketobutyrate. This is made possible through the catalyzation of the enzyme called, yes, the isocitrate dehydrogenase. So this is the first of the 4 oxidation reduction reaction in the citric acid cycle. So this oxidation reduction reaction is the one which produces energy in the Krebs cycle. And all of these reactions use enzymes which are known as the dehydrogenases. So the dehydrogenases are the ones responsible for the oxidation reduction reaction. So that is the main job of these dehydrogenases. But what does this dehydrogenases do? And how do they produce energy? Diba? Why can't the acanatase do the job? So let us discuss the job description of the dehydrogenases. So from the term itself, there is a removal of hydrogen. So as you can still remember, so if you still remember, the removal of hydrogen is also known as the oxidation. So the hydrogen that is removed from one molecule must be passed on to another molecule. So the oxygen that is released must be passed on to another molecule. So this another molecule that will receive the hydrogen molecule gets reduced. So on that, it's basically the role of the dehydrogenases. The concept is that they remove hydrogen from one molecule, oxidizing it, and in turn, it attaches hydrogen into some other molecules, leading to its reduction. So that is the process known as the oxidation reduction reaction. So I hope that is clear how the dehydrogenases do their job description, diba? Okay. In the case of the step 3, the hydrogen removed, diba? So in the case of the step 3, since there is dehydrogenase, there is an oxidation that takes place. So now that we have fully understand the job description of these dehydrogenases, so in the case of step 3, the hydrogen that is removed from the isocetrate is attached to the, anyone? Yes, to the NAD, making it, yes, NADH. So that is also a product of the Krebs cycle. So that is our first NADH na product. Addition to this reaction, a carbon atom is removed from the isocetrate in the form of what? In the form of carbon dioxide. So this leads to the formation of a 5-carbon compound called the alpha ketoglycerate. So if you look at the complete process of this step, oxidative so yes, that is the 5. This alpha ketoglycerate is a 5-carbon compound. It's because the carbon came from, yes, it came from the isocetrate. So if you look closely at the complete process of this step, oxidative decarboxylation takes place. Oxidative in the sense that a hydrogen is removed from the isocetrate, and decarboxylation as in the sense that a carbon group is also removed from the isocetrate. So the conversion of isocetrate in the alpha ketoglycerate is followed by a second decarboxylation reaction. So again, there is a second. There are four oxidation reduction reaction that will take place in this Krebs cycle. Okay, so now that we are done with step 1, 2, and 3, the next step that I am talking about will be furtherly discussed by Mr. Coseniero.